Let's see if this works. Are you able to see? What can you see? Let's.
Yep, I can see your editor view.
Okay, and now?
Yep, that's perfect. Yeah. So, fantastic. Welcome.
Thank you. I cannot see you guys though. Hold on, is there?
Yeah, that's a problem with Meet.
Oh, okay.
Zoom
All
at
right.
least has a little screen to the side that shows you yourself, so or everybody else, just so you have
Yeah.
some presence. But here, it's kind of odd.
Okay.
You're supposed to have a second screen, which most people don't.
Oh, let me see if is that true? Is there something creative I can do here? Let's see.
Oh well, you can join on your phone. Any other
Uh,
device works.
yeah, I'm just going to join on my iPad
Yeah,
as
yeah, that's
well.
perfect.
And then if that doesn't break everything, at least we know the talk works, so we're not too
Yes.
worried about breaking everything.
Yeah, and you join with companion mode, so it's like, for example, actually, that's how people watch slides alongside a talk. This way, your video and audio is not going to go through the iPad.
Yeah. This is an education in using a new platform.
Yeah, and you'd think they would have had enough years to get that speaker use case, but
Yeah, but I think it's you have to invest in what makes sense. If the majority of the
oh yeah,
world
yeah, yeah.
using, okay,
Yeah, they decided what kind of use cases they wanted and just like focused on that.
NIY, GTPK, and it's called no. Do I want? How do I join? I don't want to share screen. Joining info. Let's see if this works.
Cool, I can see you. If you turn the camera off, then you should be able to follow along. And right now we see two of you. Perfect. All right, fantastic. So then, yeah, let me start recording, and some people might trickle in if they want to ask live questions. Otherwise, I'll just, people might ask questions through me, or ask questions during the call. Of course, it's the recording. So 1 second. Okay, so when it starts, and then I'll do a really, really brief intro.
Okay, I'm just going to make sure my phone's muted so things don't go.
Good point.
Usually it is, but it's better to double check. Okay.
All right, cool. So yeah, hi everybody. I'd love to introduce Padmini Rangamani from UC San Diego, and she studies, simulates membranes and like the components of cells that, I guess at the finest level. In this case, in this talk, focusing on the biophysics of neurons. Yeah, welcome, Padmini.
Yeah, thank you very much, Greg, for the invitation, and I'm looking forward to sharing some of the work we've been doing. Just as a brief sort of background, I put together these topics with the hope of stimulating more discussion with all of you, so I don't have to go through everything. If there's something that just merits, a little bit more chatting, we can do that as well. So I am a biophysicist by training. I trained classically in engineering and then in biology as well. So I am generally, my interests are very broad in the sense of the kinds of problems I like to work on, but very focused in the world of cell biology. And I usually like to show this slide here to showcase how many complex biophysical problems one can think about in the world of cell biology, whether it is the cytoskeleton and soft matter physics or membrane trafficking or organelles and things like that. And of course, these days, a lot of people are very familiar with viruses and infectious diseases, and how these get taken up is a focus of membrane trafficking, which is a good portion of what my group does. But today I'm going to be talking about primarily structures of a lot of these cellular organelles and what computation and biophysical simulations can help us draw some insights into. So I've organized this talk to sort of give us a little bit of biological background to have a sense of why we care about these problems in terms of what are dendritic spines and why we care. We all care because we are trying to understand the human brain and learning and memory, and then we'll talk a little bit about using physics and geometries to address some of these questions. We'll switch a little bit into neuronal mitochondria because there's a lot of data there, and then how we use these for simulations. So what are dendritic spines and why we care about them in the context of neurons. So each of our synapse has like a presynaptic terminal, and then there's the receiving part that is receiving the neurotransmitter signal in the neuron. And these are usually, many of these postsynaptic parts are these little dendritic spines. What they are along a dendrite, which is a protrusion from a neuron. A neuron is a very large cell. These dendrites have these little protrusions that are often seeking a partner axon or a presynaptic terminal, as they're called. And these dendritic spines, these little protrusions are called dendritic spines, very well characterized with a neck, a well-defined head. They have these purple regions here that are marked that are known as the postsynaptic densities, which house a lot of these receptors. And recently, over the years, there's been a lot of, I'm sure some of you at Google are familiar with the effort at connectomics of trying to identify all the neurons that there are in the brain of different organisms. But if you look at a single neuron, you might see protrusions like this, depending on what type of neuron it is. Now, what is fascinating about all of this is you also see these little internal structures. You can see the yellow here is what is known as the endoplasmic reticulum, and you can see it's very highly organized structure. And then under there, you can also see the mitochondria, which are obviously very important. Why do we care about this? Anyway, this is just to show you how the 3D structure is organized. Every time you learn something, you form a memory. In fact, in the 5 to 10 minute time scale, these spines actually grow in size when you sleep, and there is a reset. The spines can shrink, and you will see a loss of spines in neurodegeneration. You will see a loss of spines in addictive conditions. And so these are actually sort of these biophysical computational markers for how our synapses are functioning. So if you look inside a single spine, and this is a cartoon that we've made up because we were trying to organize a lot of our thoughts into what's happening in there, you will see a lot of complexity. You can imagine if any of you are in the world of computational chemistry or, molecular dynamic simulations, there's a lot to play with here. You have a cell membrane, and you have all of these channels and pumps. These are characteristically represented by, Hodgkin-Huxley or Fitts-Nagumo equations, depending on what we are trying to capture. There are fluxes that are coming in from the outside. There are neurotransmitters. And I told you just now that these spines actually change their size and shape, and that is driven primarily by the rearrangement of the cytoskeleton, which are these actin molecules. So depending on your interest, there is a lot of biophysics that one can do. And, the world of cell motility has benefited a lot from these sort of physical models of cytoskeleton rearrangements. And more recently, we are foraying in with the same sort of biophysical approaches into dendritic spines. But to be able to do things like that, you run into some challenges. Historically, systems biology has a lot of work done, mathematical modeling, network modeling, math biology, big contributions. But where sort of some of the more interesting technical challenges come into play are what geometries are you characterizing, whatever it is that you're interested in. So you may be interested in, for example, solving the Hodgkin-Huxley equations, but in a spatial format. You may be interested in just looking at calcium channels and pumps. You may have other interests like electrodiffusion and things like that. But where things get very challenging for us is thinking about these realistic geometries, which we'll talk quite a bit about. And you can get quite a bit of insight from idealized geometries. It is not uncommon to think about the cell as a sphere or other organized structures to get some leading order analytical insights. But if you actually want to get insights into some of the more details, you would need to go on those realistic geometries. So that's basically where much of my talk is going to be focused on. And if you're able to do that, you might get some insight into how these dendritic spines work. So how do I think about But what am I actually simulating? What are the biological processes? So I can talk about the geometry a little bit, but I need to set the ground for what are the processes that I'm actually simulating. In the world of learning and memory formation, from a neurobiological perspective, there is this process known as long-term potentiation, about a 1 hour time scale. And you can see that based on a lot of experimental work, we have broken it up into different steps. So if you are a channel aficionado, a biophysicist of channels, you're working in the tens of milliseconds. If you like calcium signaling and such, you're working in the tens of seconds. There are various other biophysical organizations that take place here over these different timescales. And the reason I'm sharing that with you is in the hope that we can actually think about some interesting long-term high-performance computing for physical simulations. Just as a detour, my interest in this comes from a biological perspective, but also I see opportunities. For example, those who are familiar with physical simulations for animation or, the Pixar, DreamWorks types of movies, the sort of realism that comes in those simulations, right? Can we capture some of that at a microscopic level for biology? That is sort of one of the bigger interests that I have in my group. So I'll show you some of those efforts here. All right, so we are going to sort of focus quite a bit today on some of this length scale, this time scale simulations, because calcium is the key indicator of a lot of what will happen downstream. No calcium, no dice, no long-term potentiation. So we're going to focus on that. And so how are we going to do this? You can write for a lot of these reactions, because of the work done by biochemists for the last 70, 80 years, you can write reaction rates. So you can take those reactions, you can assemble them into a system of ODEs, and you can do your classical stability analysis or dynamical systems analysis, no problem. But if you actually wanted to solve it into sort of the spatial framework, what you would need to do is, in the deterministic world, of course, you would write your reaction diffusion equation. And one of the things that I won't be going into detail today, but becomes very important, is figuring out your boundary conditions. In graduate school mathematics, we are all taught about the Dirichlet and the Neumann boundary conditions, but much of the reactions that take place in these systems are all occurring at the interface of the bulk and the surface. So you end up getting these mixed boundary conditions, not quite mixed boundary conditions, but Robin boundary conditions, where the fluxes at the interface, so your reactions rates, are balanced by some diffusive fluxes. So this leads to interesting computational challenges, and I will also identify some need for some high performance computing in that case. So that's sort of setting up the background, and I'm going to sort of show some results that start with idealized geometries to just set our framework and how to think about it mathematically, and then move to realistic geometries. But before I do that, any questions on the biology so far, just to sort of pause, because I'm not sure the audience generally has worried so much about the biology, so I can quickly answer any questions.
Yeah. So actually, it would be good to understand the course idea of the process involved, because I guess you're focusing on just the spine and the flow of calcium, but I guess why? And
So
how does it change over time and over space?
So the reason, and what I'm going to show you today, I focus on calcium in the spine and time and space, is because it is the very first thing that comes in. So if a neurotransmitter is released in the presynaptic space, and that neurotransmitter, so glutamate or whatever you've heard of, right? Dopamine, glutamate, they're heard of, they're released, and they have to bind to their partner receptor on the receiving side. And those receptors are usually calcium channels. So those calcium channels have to open, let in calcium, and that then sets the stage for everything else that happens. So this is pretty well known.
Okay, and these are, I guess, spatially, these things vary throughout the length of the synaptic spine or around the diameter, because what's the geometry?
Yes, so the receptors are now known to be very spatially localized. They form nanodomains. The calcium stores, which are the endoplasmic reticulum, are known to be present only in some cases. So there seems to be a lot of structural fine-tuning that spines seem to do to be able to get some spatial control of calcium. So here's the way I would think about this question. Calcium, small ion in water, diffusion coefficient is about 280 microns squared per second or something. So there is no reason for this small ion to be spatially localized. It's free to diffuse. You are never going to get a meaningful gradient of any sort that can give you then local activation in a cellular context.
Mm-hmm.
So if that is the case, then how does this actually give you localized activation? Why is it that only some synapses fire and not all of them? Those are the sorts of sub-questions one is trying to understand. Is it just all chemical organization, or does any of the physical organization play a role in a cell?
Okay, so the question here is, if I'm kind of trying to summarize, is you normally have this giant tube, which is the dendrite,
yeah.
And normally you would think of it as being pretty much the same, and all dendrites should be doing pretty much the same thing.
Yes.
But the idea here is, depending on the state of learning, the spines in some dendrites may change their morphology, become bigger, smaller, and that affects the functioning of that particular dendrite, which is different from the other ones. So you're looking at the way that these spines control, well, the diffusion of like calcium channels and calcium and so on. Got it. Thank you.
And so this question that you just summarized so beautifully is like pretty well accepted in biology. Like not all spines are the same. The question then becomes why, and then you're limited by what you can image and what you can measure. And that's where computation comes in.
Got it. Thank you very much.
Yeah. Okay, great. All right, so let's try to see if we can put some of these differential equations together in idealized geometry. So you can do a lot of simple, engineering. I won't go into this stuff in detail. I just want to show you how our thinking has evolved over the years. So you can start here. You can write a reaction diffusion equation, lump all your reactions in a certain time scale. You can prescribe a flux at one boundary. So this is at the external boundary, and you can prescribe a flux at the internal boundary. And when you do this, you can basically get various harmonic functions as your solution as a function of geometry. So what is the question that we're asking here? We are asking what we thought at that time was a fairly simple question. I can have a spine head that looks like a sphere, or I can have a spine head that looks like an ellipsoid. Given that, what does this set of differential equations give me in terms of, spatial gradients of calcium? And the answer to that is you get basically various harmonic functions. Here's my geometry. The solution to this equation, which is basically the Helmholtz equation, depends on your various geometric features, all else kept constant. If that is the case, then spines that are of different shapes should then give rise to different calcium features. So this is basically where we started very naively now many years ago, just coming from can we write a differential equation for this perspective? And what this does for us is allows us to then start adding detail. Now, if you have worked with any sort of very well characterized biological problem, by which I mean there are so much experimental data that you are you come into a field at a time when you can no longer write simplifying equations, which is, I think, the case for calcium signaling. Then you have to consider the kinetics of the different receptors that are involved, the different pumps and the channels and so on. So the biochemistry starts to actually play an important role. The geometry plays an important role. And then finally, transport, with diffusion and these bulk surface interactions also starts to play an important role. So with that said, we actually, I don't know why this is doing this. Hello. A lot of detail. We actually started to build up a lot more of these, channels and pumps, the transport factors, and then the geometry of these spines, and basically then elaborated this question into what happens if I change the volume of the spine head. So what happens if I change the shape of this spine head? If you're mathematically or physics inclined, you are not surprised at these results. I think I want to say more about why we chose these questions. Why we chose these questions was because the electron microscopy data was telling us a lot about the size and the volume of these spine heads. So you had these mature spines that were bigger, that were, more resistant to neurodegeneration. You had these smaller spines that were nascent, that were more seen during a learning event. And then if they became bigger, and then they became more stable and things like that. And the key thing out of what we got out of this study was that your calcium dynamics really depended on the spine head volume, not surprising, but also on where you put these different receptors. So you can put your NMDA receptors at the head. This is pretty well known that they are at the head, but the voltage sensitive calcium channels are everywhere. So we were reading the experimental literature. You can see we published this in the Journal of General Physiology, and we were trying to make connections as we were learning the field to go into that. But let's go into talking about realistic geometries. So let's talk about that. So what do we do with realistic geometries? And this is where I'm hoping that, we can have some conversation that might excite all of you as well. And what is the challenge with realistic? What are the advantages with realistic geometry? The advantages with realistic geometry is these days you have a lot of data sets. So here is an example. So let's look at this first. I'll explain what's going on here, and then we'll see what we can do with it. This came out in 2017. FIB-SEM. SEM is scanning electron microscopy. FIB is focused ion beam. It turns out that this is important for a theoretician like me to know, because not all electron microscopy can give rise to data that is of this 3D resolution. What you're looking at, X, Y, and then you're going to go through in Z right now, and all I see is a bunch of gray lines with, a lighter gray background. But all those gray lines are different cellular membranes. And what is fascinating about this FIB-SEM data, which I have learned since, is that this data is actually pretty high resolution in Z. In EM data, it is no longer that hard to get very high XY resolution. You usually get 4 to 5 nanometer XY resolution, but getting Z resolution remains a major challenge. So if you look at this data set, what you can now do with this is actually start tracing. So
are we looking at a single spine?
We are looking at a section of, it's not a single spine. The single spine is now what's playing with the teal line outlined in it. What you're looking at is just a section of like brain tissue. So you can't, I can never tell what is what. Someone's got to tell me what is what. It's all gray to me. So someone has to tell me on, okay, over time you start learning a few things, but I would not trust my own judgment on what is what. But so what you then do is you sit down and you trace these lines and you register the images in each Z-plane so that you can track them. So this teal, becomes this. Okay, and then you can see. So let me tell you some things that I have learned over the years. So if you actually watch, you can see my mouse pointer. If you watch this, that is a mitochondria, by the way. And you can see, if you just keep your eyes on this and not on the lines that come off, you can see the mitochondria disappeared as you go in. But that's the
yellow bit, right?
Yeah, that right there is the mitochondrion. And the reason I know that, so here's another mitochondrion, it's because it is a double membrane.
We can't see your mouse, but I can see in the picture it gets yellow, and also to the side it gets yellow. So it appears and disappears as it gets thick, thin, right?
Yeah. So the mitochondria is the one that's darker gray with double membranes. That you kind of learn to identify that over time.
And presumably, so I'm not seeing much. So the permeativity through mitochondria presumably is different than through internals of this spine, right?
Yes.
And are there additional structures that it's kind of hard to see? Like, are there membranes within the spine that have different permeativity?
Yes, they do.
Cool. Okay.
Yeah. So one of the things we did was we actually so this, by the way, this tracing, the teal that you can see is basically manual tracing and reconstruction, very, very labor and time intensive. And so there is actually room for, if anybody is interested in any sort of AI machine learning type work, to do this better and faster. Right now, we are rate limited by getting these sorts of images into the simulation platform. So if you actually do it manually, and this was done by Pietro Di Camilli's lab, what we have been doing is getting these 3D reconstructions into high quality meshes that we can use for simulation. So here's the workflow. At each and every step of this, and we can just chat about this slide for the rest of the hour if that is of interest to the audience. Each and every step of this needs more computational investment. I can tell you a little bit about what we've done here. In the world of segmentation, registration, filtering, the state of the art remains manual curation. And this is a big sort of bottleneck to realizing, biophysical simulations to mimic cellular processes. And I'll show you some of what we've done in the world of mesh generation and then simulation, for especially the finite element world.
So let me ask you
You
about this
take
one.
the 3D reconstruction in its native state, and you try to run it through any sort of meshing framework. You can use your, you can use TEDGen for the volume, and any of your Blender, any mesh generator sort of frameworks. You will end up getting a mesh that looks like this on the left-hand side. If you can't see my mouse here, the leftmost mesh, which, still keeps the geometric features of the spine. So what you're looking here, the bottom is the dendrite, the thick cylinder going at an angle on the screen, the neck, and then you see the spine head. So that is the spine head, and this is quite dramatically different from the cylinder and the ellipsoids, the spheres and the ellipsoids that we were making assumptions about. The dark purple regions shaded on there are the regions where we know from experiments these receptors that we talked about exist. So that's the postsynaptic density. If I took the mesh that is on the leftmost panel and tried to do any sort of finite element calculation with it, it would just not converge and it would fail phenomenally. And so we spent a few years working on mesh generation while preserving the geometry. So this is geometry adaptive mesh generation that then gives you the second panel on this slide, where you can now see the triangles are more or less equilateral. It's a much better conditioned mesh, and it is much more adaptable to any sort of finite element calculation or any other simulations one would want to do. We can do that similarly for the internal structures of the spine. So the yellow that you're seeing in there is now an internal organelle known as the spine apparatus. A spine apparatus is a specialized endoplasmic reticulum. And we can now condition that mesh as well. And you can see TEDGen mediated volume meshes come through. The third object on the screen is completely compatible with all sorts of finite element operations you want to do with like reaction diffusion type models. And that's a simulation that I'm showing you just some gradient from the Phoenix project. So this is basically the kind of things we want to do with realistic geometries. If we had the leftmost structure, then the rest of it we have some handle on. So how do we generate these sorts of meshes? It takes, the code base is open source and available on GitHub, by the way, for those who are interested. But just to show you panel B and C here, you can see how irregular that mesh is and how poorly conditioned it is, and then how we work our way through from panel C to panel F. It's a dramatic difference for this really small endoplasmic reticulum mesh, and you can now see the angle distribution is more or less centered around 60 degrees for the equilateral triangle, which is quite different from the original angle distribution that we had. And this is work done with Romy Amaro and Michael Holst at UCSD. Okay, so what do you do with a mesh like this? What can you get out of it? So going back to the sort of biophysical questions, I originally told you I'm going to do spheres and ellipsoids because I know how to do the mathematics on them. We actually can use these meshes to calculate the curvature of these objects. So just using simple discrete differential geometry expansions, you can write down for each triangle, you can calculate the curvature with the cotangent formula. And this is actually quite insightful, it turns out, for our biology colleagues, because now you can see where the regions of the high curvature and the low curvature are. What you're looking at here is on the top panel, you're looking at the first principal curvature for the spines. And then on the panel B, you're looking at the curvatures calculated for these internal organelles, the endoplasmic reticulum. So now you can see what was basically, in panel C, the dendrite that I showed you with the cylinder, where the regions of high curvature are and how these spine curvatures are distributed. So if you wanted to make some approximations, you now know what the closest we can get to ground truth is on these objects. And then you can take a lot of these, add more biochemistry to it, and then you can run simulations in these realistic geometries, which is what I wanted to do for a very long time. So you can take a spine like this, you can prescribe a voltage, that is the stimulus, you can see what the channels that are activated, and you can look at the calcium come in. So this is where we were going the whole time, is trying to be able to use these in simulations. And you can then do this.
As we get into these pictures,
yeah. Sorry?
Just to understand the challenge of just the previous section, it seemed like you were, the funny, actually, I guess the high curvature regions would stretch out the mesh. So now you would have these horrible jumps in your, in the dynamics you're about to be showing. So as I'm understanding now, it's that you have very heterogeneous distances between adjacent mesh points because of this, long stretching, because these are crazy shapes, which from the previous picture, the scans, it seems like all these spines are just pushing at each other and stretching each other out of shape, right?
Yes.
Because there's no gaps inside the brain. Is that fair? The different dendrites actually just push each other around and deform each other?
No, those are all true, but what we have is a single snapshot in time. So in the EM images, you don't have anything in time. So I couldn't tell you what happened before or after.
But the effect okay, so you don't get the dynamic deformation of shapes. That's not new data. But for a given snapshot, you get this crazy shape, which is hard to mesh. So you have to adaptively add more, mesh points in the high curvature regions, so that you have decent spatial resolution where the dynamics are.
Yes.
Cool.
Yeah.
And now here you have the dynamics vary presumably by shape, by skinniness of the spine, that kind of thing.
So the questions about why we care about the shape and the skinniness of the spines, of course, the argument is when you have a skinny spine, for example, this one, when you have certain amount of calcium come in, is that enough to bring in other chemical activity to make the spine grow and become a more stable spine? You can now start to ask, can you turn this guy into a stable spine, or is it forever going to remain skinny, or is it going to dissolve and disappear?
Got it. Because the calcium is going to give it that firmer structure.
Yes.
Got it. Okay.
Yeah. So here, what I'm showing you, there are two reasons I'm showing you this. One is, of course, to show you that we can now take from that big dendrite individual spines, classify them. You can, of course, measure their volumes. You can classify them according to the textbook by their shapes, and then, run simulations of this. In this case, just to be clear, these are non-deterministic simulations. These are particle-based simulations of calcium influx, where we remain true to the known biochemistry, but we are doing this in a framework known as MCell, so Monte Carlo particle-based simulations. And what you can now see here, so this panel B, the left column of panel B, you can actually see the distribution of calcium at 15 milliseconds. So you can see those little dots, they are the calcium. But in panel C, you can look at the vertical axis. The mushroom spines have more calcium, and that is, of course, volume dependent. And there are shape-dependent effects with the thin spines, the mushroom spines, and the so-called filopodial spines. And you can start to extract, again, what is the role of shape, what is the role of volume, and be able to make some forward predictions for, biological function in here. The key, of course, is not only do you have, like, these mean behaviors, but you also have a lot, the variance depends quite a bit also on the volume because of the stochastic nature. So if you have volumes that are very close to each other, so this is panel C2, you don't see as much as differences in the variance, but in panel C6, in the mushroom, the variance depends quite a bit on what the volume is because of these sort of spatial effects that one can start to see in here. So just recently meaning on Friday, we actually finished this work where we extended this to now the internal organelles. You remember that yellow structure that is very beautiful to look at. We now added the relevant reactions. That is known as the spine apparatus. We added the relevant reactions and now asking questions about what is the role of the spine laminarity, which is the number of layers of those structures that it has in sort of calcium dynamics. And we find, of course, that there are differences in whether you have a spine apparatus that has got laminarity or not. Why is that question interesting? That question is interesting because the stable spines have spine apparatus with increased laminarity, whereas newer spines don't have a spine apparatus. And then the in-between spines sometimes just have a spine endoplasmic reticulum without any laminarity. So now you can get into a little bit of these exploratory questions because we have the meshes that are compatible and because we have the modeling framework. I can talk a little bit more about the biology.
I think I missed what laminarity is.
I'm sorry?
I think I missed what laminarity is. What are these structures?
So in most cells, you will see an endoplasmic reticulum that, oh, you cannot see my mouse. So if you look at panel C on the left, if you look at panel C on the left, you see in the geometry that says idealized PM, you see the left has a little mushroom. So we are looking on the inside now. The light lavender colored structure is very smooth. But if you look on the right, it has these layers. Like they're almost folded over. So it turned out that structure, now you can see the similar one in panel C and C3 and C4. The more stable structures tend to be the ones that have these very highly folded, almost organization. That is the laminarity. And that has to do quite a bit with the stability of these spine apparatus structures. So what we have...
What are these? So are these layers like, yeah, well, what are these layers made out of?
They're all cell membranes. They're all membranes. It is the endoplasmic reticulum. It is how it is arranged.
Okay.
And it turns out that how it is arranged, so you will not see something like these highly lamellar structures in your generic endoplasmic reticulum, let's say, for a fibroblast. These are very unique to spines and neurons.
Okay.
And they seem to play quite, what we showed from our models is that they seem to be playing very important roles in sort of calcium signaling. That's the, if you remove the laminarity, you get very different calcium dynamics. So here is a good, so panel, so let's see, right, panel B2. If you look at the dark green and the light green, The light green is with the laminarity, the dark green is without, and you actually get everything else being the same. With laminarity, you get different calcium dynamics, and that seems to be quite important for spine to spine communication. Okay.
Yeah, that makes a lot of sense. Thank you.
Yeah. So the repeated theme that I'm getting at here is, your surface to volume ratio, where you place your objects in the cell, how you calculate your fluxes is kind of going to be quite important. I think that would be the sort of take-home message. And you could come at this from fairly, phenomenological models, which we know to be true mathematically, but we're also showing this in realistic geometries without making any assumptions about the geometry. So let's talk for a minute. So this part is a lot of fun. Let's talk about mitochondria. If it's okay with everyone, like let's switch gear, Craig, to talk about mitochondria.
Oh, yeah, absolutely, yeah. In fact, I'm curious why they're so huge. I thought they were tiny things, and then you showed these giant things floating inside these spines.
Okay, so mitochondria, turns out, are yeah, they're super small. Like you'll see. Let's get to this. We'll come back to some of these challenges, right? So we've been trying to see if we can do this. Let's get to mitochondria. Okay, so this is what a cross section of a mitochondria looks like. Forget all the text. Look at this. That's what it looks like. And here's why mitochondria are interesting. Well, high school biology tells you they are the energy battery of the cell and all of that, right? But practically every disease that you can think of, long COVID, inflammation, Parkinson's, deficiencies in things that are not quite explained by a blood test, like a fatigue syndrome and things like that, are all related to mitochondrial function and mechanics. So this I learned recently. So I thought that was fascinating. This is just my public service announcement. Is mitochondria are really cool. So they were like you. I also thought they were small. What is actually very interesting about them is they have a lot of dynamics. They will undergo fission and fusion. So they will actually fuse together to become bigger, and they will break to become smaller, to disintegrate. Good example is skeletal muscle exercise, which is something that we are studying right now. Fascinatingly, depending on the mode of exercise, whether you are an endurance, runner, or you just like to do boot camp and lift weights, or you sprint and you call 20 minutes and you're done with your workout, your mitochondria respond differently. And so just thinking about day-to-day activities on what we do and how we go about them, mitochondria are changing constantly. What do they look like? So this comes down to the question of what do they look like? So the mitochondria has an outer membrane in green, the inner membrane in teal here, and then these highly folded structures called cristae that you can see in one XY plane. They look quite like this. This is a labor of love, this project, just getting these sorted. Now, sometimes in a particular plane, depending on how the image was taken, the cristae look to be outside. If you're tracing the contiguous membrane, they look to be outside the outer membrane, which should not be possible. And then sometimes it is very hard to find how the cristae membrane are connected to the inner membrane and things like that, because we know they are continuous structures. We just don't know how this works. So what we have been doing for a while now is trying to assemble these. So these are images from Mark Ellisman's lab, very high resolution. The challenge with this sort of sectioning, so you have a block of cell, tissue, whatever, and as you're sectioning, you will lose about 60 to 20 nanometers in the Z direction. So you'll end up getting gaps. So you get really high resolution XY, and then you get these you can see these little tiling gaps here. And that's because the knife loses material in the tomogram. And so if you take each of these, you can get very high, each slice very high in XY, but then you start to assemble it, you get these gaps. So the way we did this, it took a whole bunch of really, really brave high school students, a few summers, to sit and trace this, right? I cannot imagine, I'm sure you'll all agree that, convincing, undergrads or grad students that tracing mitochondria for the research project is something very useful. But we have a very strong community of high school students who want to learn, who want to get in the lab, and basically they sat and traced all of this. And then we had to come up with some interpolation methods to fill these gaps and realign. And the author mentioned here, Rachel Mendelson, at that time was a sophomore in high school. And that's all she did all summer. She's now majoring in neurobiology at UCSD. And so you can see this. So this was basically, we would go to Costco, get a whole bunch of snacks, and have these, kids sit down and spend their summers, tracing these mitochondria. And the end of it, we got about 12 mitochondria traced, reconstructed, corrected. And this is my postdoc, Chris Lee, who supervised them, worked with them, and Tom Bartol, our friend and collaborator, who actually is really good with working with these young students and getting them to do all of this. So this is now what your mitochondria start to look like. And if you put it all together, put it through the meshing framework, that's the inner membrane, and that's the cristae. So it's just incredibly complex and beautiful, and nothing like our textbooks tell us what the mitochondria are.
So question, did we, yes, I did not ever see us pictures like this. Is this like first time we see this 3D structure, or is it just a, broader scale of like more mitochondria are imaged this way?
This is a single mitochondrion.
Wow, okay. So what are these structures that we're seeing?
So this is the inner membrane of a mitochondrion. These are the cristae membrane. That's where I was going with this. These cristae membrane are where the motor proteins that make ATP are. So we know the textbook picture of a mitochondria, right? There's a capsule. It looks like a fish oil capsule outside, and then there's stuff on the inside, and somehow ATP happens. The proteins that make ATP are on these membranes, the green ones. Sorry, the blue is the inner membrane, and the green is the cristae membrane. They're called the cristae membrane. And that highly curved structure is very tightly correlated with the ability of the ATP synthesis to make ATP. We have another works in progress right now where let me show you this, Greg, and we'll come back to this in a minute. We'll see why the structure is important. So what we did was we basically developed a reconstruction method, made everything, watertight meshes. And then what we can do with this is characterize these mitochondrial shapes where you get these outer membranes, inner membranes, and the cristae. We'll come back to curvature in a minute. What can you do with this? I think this is what I'm trying to explain. So here's the green membrane that we saw earlier. And I like that image on the left because it shows you out of plane how the mitochondria are arranged, right? So they're so used to seeing those grayscale, you can see out of plane. What we know from metabolism and biochemistry is where all the ATP synthesis are. ATP synthesis make the make the ATP, excuse me. And the A and T are the nucleotide transporters. They export ATP from the inside of the mitochondria to the cytosol. If you have mutations in the A and T, which is in red, you cannot get the ATP from the inside of the mitochondria to the cytosol. If you have a mutation in ATP synthase, you cannot make ATP. So two parts, you got to make it and then you have to get it out. And other studies have shown that these molecules are located in very specific locations. So if you look at now the green cristae membrane that are shaded in red, those shades are consistent with the curvature-dependent locations of the ANTs and the ATP synthesis. Okay, so are we good so far on where we're going? So that's your mitochondria. That's why that green membrane is important. It's way on the inside. You break the green membrane, no ATP. Just very coarsely.
So the story is ATP is made inside the mitochondria, ANT ferries ATP from inside to outside. The amount of ANT depends on the internal morphology of these shapes.
The amount of the location of ATP synthesis and the ANTs depends on the internal morphology.
Got it. And that you get from other studies.
Yes. Ergo, our prediction is how much ATP you make and transport to the cytosol will depend on the crystal geometry and the mitochondrial geometry. You can imagine that if you're able to look at the morphology, you cannot simultaneously look at the ATP production in many of these experiments. What we're doing is taking those geometries and then predicting which ones are able to make more mitochondria, more ATP or less ATP. And going back to what we did with those 12 mitochondria, the key thing we did was using our meshing, we were able to characterize them in terms of their curvatures, whether they were globular or elongated. So you can start to infer shape-function relationships on these mitochondria, if that makes sense. And this can be very useful as a predictive tool for the many conditions in which mitochondria are involved. Okay, so what I want to do very quickly in the interest of time is actually show you, I'm going to show you, show all of you some really cool movies, and then we can come back to the biology, because I want to talk for a minute about high performance computing needs that we have in here. Is that okay? There's movies.
Yeah, that would be great. Thank you.
Yeah. So everything I've told you has been building up towards this. Or we can do spines. We as long as we can get enough, motivated students to trace these geometries, we can do spines. We can do ER. But I only showed you like tiny subsegments of cells or like a single mitochondria. And what I'm going to show you here is trying to get a whole cell simulation. It's like the pipe dream, if you will. And all of these blue structures here, they are the endoplasmic reticulum from a Purkinje neuron, which is a very large neuron in the cerebellum. And you can see what now the endoplasmic reticulum structure looks like. I couldn't tell you exactly what it is. It's a bunch of like tubular structures. And the first thing you'll notice is it's very different from those laminar structures that I showed you earlier in the dendritic spine. And we are trying to get calcium simulations running on this entire geometry. As you can see, mesh generation is a challenge. We have some preliminary work on it, but let me give you, I'll skip the math here, but I want to give you some highlights. This is the highlight I want to give. If you take, let's orient everybody here. Here in the region that says nuclear region, that's the cell nucleus. We don't have that data. We don't know what's in there. The green is the cell membrane. It's the plasma membrane. Everything that is in that gold color in there is the endoplasmic reticulum. And the gray is just the volume in there. Okay, so that is a fairly large cell. And it took us a lot of work. This is after the EM was done and reconstructed. It took us a lot of work to get this into any sort of reasonable mesh. And you can see the details of the mesh there. That is about 62 million tetrahedra. And the mesh, the volume meshes are more inside the endoplasmic reticulum, which is that gold yellow structure. There are 37 million inside because it's a very thin, narrow space. So you got to get your volume meshes done really well. You can see that in the inset. And 25 million in the gray space, that is the cytosol. Okay. The surface triangles in total are like 11.6 million surface triangles that we are simulating. And for reference, this is only 1/2 of a whole cell. So a lot of our initial challenges were even getting these meshes, getting, I run an academic lab. We are run-of-the-mill operation. We try to do a lot of things on desktop. Maybe we get ourselves a nice server if the grant has equipment money, but we would need to go through Exceed or some such place to get supercomputing. So it's not that the resources don't exist, the scale of my operation and the scale of resources. So this, if you put that in this context, this is a huge achievement, even be able to mesh this in this context, okay? What you can do now, I'll show you these movies and then we'll talk. You can actually, if you have the mesh, what is the problem? The problem is now when you start writing all those reactions that you care about for calcium and you want to keep any sort of biological realism, you now have two problems. One is you're going to make a lot of errors with various kinetic parameters and things like that, just manual intervention. So you want to minimize that every time you set out to do this. The second thing is, remember I told you those flux boundary conditions and things like that? Every boundary, every interface has like a gazillion flux boundary conditions. So you need a way to be able to do mixed multi-compartment, row-bound boundary conditions robustly. And as much as I have used and loved COMSOL cannot handle this. We were not able to get this mesh into COMSOL. So we switched to working with Phoenix and working with folks at the Simula Research Laboratories in Oslo. And I'll show you this movie. Well, this one on the left first, that is the calcium dynamics. And what you'll see is calcium come in 5 pulses, which mimics what happens to this neuron. And that's the sort of background I'll tell you, and then we'll play it again. And then you'll see these traveling waves. So what happened here? So what happened here is that's the first pulse in response to an electric signal coming from the outside in, so no surprises there. Goes away very quickly because calcium diffuses. Nothing to see here. Second pulse, third pulse, and now there's a hotspot over there. Why did that happen? That happens because in our cells, there are ways in which there is feedback. There is this thing known as calcium-induced calcium release. That's what keeps our heart beating, our neurons firing, and there are other mechanisms, and that is coming from this endoplasmic reticulum. Okay, so the endoplasmic reticulum is releasing the calcium. All of this biology, very well known, incorporating it into these models, extremely, extremely challenging to get the nonlinear mathematics right. So now you can see after the third pulse, there is a little hot spot on the bottom left there. And now you go there, it's growing because that is coming from the inside. See where the red is? That's coming from the inside of the cell. And the fourth pulse came and it grows, it disappears, moves around depending on the location of the endoplasmic reticulum. And now it starts to spread. Now you start to see traveling waves come from the inside to propagate these fast pulses to becoming more longer term events. And there was the fifth pulse I just showed you. And there you go. You let it go and then it just does its thing and dissipates. So these are all the sorts of, this movie simulation took a month to run at the supercomputing at SDSC that we had some time on. So just to give you a flavor for where we are going with these sorts of biophysical simulations, we have a lot of analysis to do on what this means in terms of physics, but we also recognize that we have only gotten to 100 milliseconds in our simulation. We want to get to so much more, and 100 milliseconds took us like a month. And of course, Justin Laughlin, the student who did this, took on so much work. But this is the bulk of what I wanted to talk about. I am happy. Here's where we want to go, just for all of us to think about. We really need to catch up with the experimentalists in terms of what data they're able to produce and how we are able to use it. High performance computing, and then we need to be thinking about deformable structures, because right now all of these are static structures. They're not changing, they're not responding, they're static. So that's the summary of it. I will stop here. We are just on the hour, but I can stay for a few minutes if you guys have time. Everybody who did all this work, my group and my collaborators. So I will stop there and be happy to take any questions.
Yes, thank you very much. I think Roy, you have a question.
Hi, can you hear me?
Yeah.
Great. If you can go back a slide or two. One. Yeah. So in this simulation, you really take the most fine-grained resolution that you can use to, I guess, see the point of how do you call it? Triggering, the point at which the calcium is released?
Yes.
Okay. And what would happen if you take a coarser tessellation, but in which you have a compositional feature? So you have two materials in here, right? The one creating the pulse and the one which is the ether through which the calcium moves. So if you create rough, coarser tessellations, but take them to each cell, give a float value between this is entirely type A material, this is entirely type B material, would then there be a way to generalize the diffusion equations you're using so that you don't have to do so many?
Yes. So that is the hope. So that is our goal as well. But we did not want to generalize in an ad hoc manner without knowing what we were missing. So one of the longer term goals is to be able to come up with some sort of a homogenization algorithm, so you wouldn't need to always simulate every sub detail. And we are hoping that these fine-grained simulations will tell us what would be the important pieces that you need to keep and what would be the pieces you could potentially homogenize or discard. We don't know that yet.
Right. So, for example, you would run a few of these very fine-grained simulations and then try to create a module that can work on coarser cells.
Yes. So maybe I can show you a little bit of an idea I have on that. So the idea would essentially be not these. So the idea, one idea we have here is actually that in some regions of the cell, it's hard to see here, but let me point it out. So in some regions of the cell, we see these two structures are parallel, and they seem to have a different behavior than in some regions where they tend to be more perpendicular. So that at the very minimum, you can start to say if the ER more parallel with the PM, you will see this kind of a, either a traveling wave or a hot spot in the ER perpendicular to the PM, see a different kind of behavior. So that's like one of the ideas we have that we have to test out a little bit more.
Yeah. Thank you. Very interesting.
Yeah. And Farouk?
Hi. Yeah, thank you. That was a great journey through multiple layers of biology and physics. I had one question on, you mentioned the high performance computing needs that you had. Do you have, or could you say some of the names of the software you're using for a simulation, and maybe kind of a rough estimate of the computation requirements for simulations such as these?
Yeah, so we are right now, these simulations, these PDEs, we are running all of them using Phoenix. We've been working with folks. So Phoenix is the open source Python-based finite element software. We've been working with folks at Similar Research Lab, some of whom who originally wrote Phoenix to work on this. Our hope is to be able to eventually add even stochastic differential equations, which are harder to solve than deterministic ones. In terms of the specifics of the computational needs, I will email you back on that, because for each of these movies we showed, we used different setups. In some cases, if for some of those M-cell simulations, you can do quite a bit, at least test case runs on a desktop, but then once you want to run 200 iterations for stochastic calculation, you do want some supercomputing time. So I can I can follow up with you on some of these details. I don't have them off the top of my head, unfortunately. Sorry.
Sure, sounds great. Thank you.
Yeah.
Yeah, and I'm also wondering how, if you want to start making neural surrogates of something like this, do you need to? It would be bad to run the whole thing, but maybe you can do small, like,
Yeah.
Segments in time and space, and run it a bunch of times to say, here's a summary of one of
Yeah.
these substructures.
Yeah, and then I think I might stop sharing my screen so I'm not looking sideways at all of you. I think as some, parallel computing needs, definitely. Right now, we didn't know what we were solving until recently, so we haven't parallelized it. I mean, and whatever we solve has to actually make mathematical and biological sense. So we've been doing a lot of convergence tests and mesh size and time stepping, and so we're still in the world of computational mathematics. So we need to move it up a little bit more to computational, like better performance. We haven't worried so much about the performance metrics from speed and efficiency. We've been focused on the mathematical performance.
Okay. Oh, and one thing for validation. So I notice you have a bunch of things like, it seems that if you account for these structures, then results notably change. But do you have biological data that validates your predictions?
Yes. So a lot of, biological data that at this point constrains our model, validates our predictions, not yet, because to be able to measure these calcium dynamics in subcellular, it's hard because a lot of the fluorescent sensors will just light up the whole cell.
Right.
But our time courses that I show you are usually pretty constrained against known experimental measurements. So we are not off by, seconds, and the peak values and the concentrations definitely are all validated.
Mm-hmm.
So that's where we've been, I would like to think very careful in building these models. So the one reason to choose calcium dynamics as one of the sort of goalposts is there is a lot of mathematics done in calcium signaling. Every channel, receptor, probability, density functions, so those are all pretty well known. So you're not looking for new equations on top of new geometries. So you're taking established equations, putting them in new geometries and asking, will my results change? And of course, mathematically, we know it'll change. But then when you start seeing interactions like these traveling waves at the end, you're going from a single spine to a half a cell. Those, I don't think we could have predicted. Where will you have the hotspot? Oh, it's close to the nucleus. Does that mean something for nuclear organization? So you can actually get into very interesting biology.
And then you need a model for the nucleus, you need a model for presumably adjacent cells that are giving the calcium.
Yeah, exactly. So you can actually build up quite a bit.
Okay. So, well, yeah, fascinating. Thank you very much.
Oh, yeah. Thank you very much for having me. Yeah,
absolutely.
I'm happy. Greg has my email, so if anybody wants to follow up or chat more, I'm happy to, always.
Oh, yeah, and please send me your slides. I'm publish them so everybody can follow along, and maybe you can get contacts that way.
Yeah, wonderful. Thank you so much.
Thank you. Have a great day.
Thanks.
Bye.
Have a good day. Bye.